This module is the first part of the Basic Inference for Microarray Data lab. We're going to build off the two strains of mice data set, which Rafa described in the inference modules. So we have strain 0 and strain 1, and we're looking for differentially expressed genes between these two strains, which have been sequenced by microarray. So we're going to use the MA pooling data set, which is in the DAG data package. And I have the two lines to install this DAG data package from the genomics class repository if you don't have it already. Then we use the data function to load in this expression set, which is MA pooling, which stands for microarray pooling. The data set has a number of samples of these two strains, and it also has pooled samples, so pooling the biological replicates and then having technical replicates of those. So the phenotypic data table for this expression set has a row for each microarray and a column for each experiment. So experiment A10 is present in the first row, and it's also present in the second row. So the second row has a 1 for A10 and a 1 for A11, which means it's a combination of the A10 and A11 experiments. So this is the pooling. But we're only interested, first of all, in the samples which are only coming from one experiment. So I'm going to first make a plot of the phenotable, and then we'll figure out how to extract out the single experiments. So I'm building this function myImage, and I'm going to first define a function flip and transpose, flip t. And the reason is that if you use the image function on a matrix, it'll show a picture of that matrix, but it'll be flipped around. So I'm just redefining this so that the image which is produced looks like the matrix which you see printed out by R. So here is the picture for the phenotable, and it's the same as the matrix I printed out before, except it has a black square instead of a 1 and a white square instead of a 0. So we can see the block pattern, and so we're interested in finding the rows where there's only a single black block. So we want to get rid of these stripes. So we can take the row sums of this table, and we're just interested in those rows where the row sum equals 1, so where there's only a single value in the row. And then that gives us these values. So these are rows of the expression set which are not pooled. And then we can furthermore remove the replicates, because there are some of these which we have multiple replicates of the same experiment. And for the analysis we want to do further down below, we just want one sample per experiment. So we don't want the technical replicates. So for instance, A3 TR1, A3 TR2, these are technical replicates of the same experiment. So we can use the grep function to find these, and then the minus to remove those samples which had the TR in the name. And now I'm making a subset of the expression set which only contains these individuals and as single replicates, so no technical replicates of any of these. And if we look at the plot, we see we have what we want. So we have a single dot for each experiment. So now I'm going to make a factor which is built off of the column names. So the two strains are given column names with an A and a B, and I'm going to turn that into a numeric, so a 0 and a 1, and factor. So now we have everything we need to do some inference. I want to stress that the values we're using are normalized gene expression, and we're actually going to talk about normalization in the next week. But let's just continue given the fact that this expression set has already been normalized. So let's look at the two genes which were also shown in the lecture, and I'm just going to show what's called a strip chart, which shows the gene expression values, these normalized gene expression values for each sample split by group, and I'm showing you for two genes. So gene 1 has largely overlapping values, while gene 2 we can see a clear decrease for group 1 in gene 2. So what I provided to the strip chart was a list. I used the function split to split the expression vector for gene I across group. And then I gave this list to the first argument of strip chart. So now we can go ahead and start computing t-tests for each gene, so for each row of the expression set, and we're going to test to see if these differences are significant using a t-test, a two-sample t-test. So the library gene filter, if you don't have it already, you can install it with BioC Lite, and we're going to use this because it has a function row t-tests, which compute very fast t-tests or f-tests along the rows of a matrix. So we're going to use the row t-test function and provide it a matrix and a second argument, which is a factor, which in our case is the group. And we can see we have a data frame. So we have a row for every row of the matrix we gave it, and we have these three columns, statistic, DM, and p-value. And in the help for row t-tests, it tells us that we have the column statistic, p-value, and DM, which is the difference of the group means. Let's focus on the first row of the data frame returned by row t-tests, and we can show that if we take the first row of the expression set and just the values for where group equals 0, we take the mean of this, we get 10.02, and if we take the mean of this and subtract the mean of the other group, we get negative 0.09, and this is the same as the value which was given back by row t-test. So it's taking the mean of the first group and subtracting the mean of the second group. And it's important to remember that that might not be the same difference in means as provided by other functions, such as the functions in lemma. These would give you the second group minus the first group. So now I'm going to use the t.test function, which would operate on a single row, and to show you that if you specify that the variances are equal of these two groups, we get the exact same p-value from row t-tests, except that row t-tests is much faster. So if we look at the p-values which were provided for gene 1 and gene 2, we see we have a not so low p-value for gene 1, and we have a very low p-value for gene 2, so 10 to the minus 7. And that makes sense because we can see clear separation of these values for gene 2. So you can make what's called a volcano plot, which plots on the x-axis the difference in means, and on the y-axis the negative log 10 of the p-value. And so you see that when the difference in means is small, the negative log 10 p-value is small, so big p-values. And when the difference in mean goes out to 2 or 3, you see that the negative log 10 p-value goes up a lot, which is a very, very low p-value. And you can use the identify function, which is useful if you're interested in calling out individual points on a plot, and it will return to you the index, the number that plot is in the data. So you can go over and then point, and in RStudio it'll give you a little blue arrow, and then click Finish, and that returned the index of TT, which is that point, which is 14925. It writes it on the plot. So in the part 2 of this module, I'm going to compare the t-test with a non-parametric test, and I'm also going to show you how you can do the t-test also using linear models.